Hello everyone, and welcome to today's webinar, Utilizing the Gibco CTS Rotea System, a versatile closed manufacturing system for cell and gene therapy development. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Gibco by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Ann Santos, Cell and Gene Therapy Senior Business Development Manager at Thermo Fisher Scientific, Emily Benz, Process Development Engineer at ViaCyte, and Eric Dubofsky, Biomedical Engineering Master Student at Cal Poly San Luis Obispo. Our speakers will now begin their presentations.
Okay. Thank you so much, and I would like to welcome you again to this webinar, and thank you for taking the time to get to know more about our Gibco Rotea Counterflow centrifugation system. And before I get into the advantages of the Rotea, I just want to say the four main pillars that Thermo Fisher can support you in your cell and gene therapy activities. So first and foremost is quality and logistics. Of course, we take this very seriously because we know that failure to manufacture equals failure to treat. And a lot of patients are relying on both you and I to transform their lives. So three things: management of supply chain and logistics. Make sure that you have quality of supply, make sure you have continuity and assurance of supply. Also, we have redundancy GMP manufacturing. Again, we want to make sure that you manufacture your therapies patients. And along with that, of course, is I'm going to click off my camera here, but also along with that is advanced analytical tools so that you know if your therapies are safe. Second is speed to market and innovation. We want your therapies to get to the patients, but without losing that quality. Regulatory, very important that we support you on the compliance, documentation, all of those things, because as you know, if you don't comply with these regulatory requirements, the FDA or the regulatory agencies will not let you get those therapies to the patient. So we also help you with clinical trials risk management. Again, fourth, and this is the cost. And to me, this is very near and dear to my heart. I come from an immigrant family, so I don't come from a rich family. So when I decided to become a scientist, I wanted to impact the world. And if we can't make these technologies affordable and accessible to everyone, that's just going to be a travesty. So we want to work with you so that we can lower the cost of goods. And in order to do that, we have to come up with scalable, closed, automated, and modular systems like the Gibco Rotea system. So why do you need it to be closed? It's because you will lower those costs if you have a closed manufacturing process. This image right here, the first one, that's open and manual processing, that gives me heartburn and heartache because you don't want that. Look at how many machines you need. And then the second one is closed and automated, yes, but it's integrated incubation. So the machine itself will tie up your entire manufacturing process. And the third right here is our vision and the way we've designed and engineered our systems and our manufacturing process. It's closed and automated with centralized incubation. So you see here the manufacturing footprint, it's smaller. And then I want you to check this right here. These are processing systems that you will need for a typical CAR-T therapy, 2,000 patients per year when you get to commercialization. And for the integrated incubation, you'll need 78 of these machines. But for closed and automated with centralized incubation, you only need 7. So can you imagine how much cost you're already going to save with just this closed and automated with centralized incubation process? And how do you do that? And it's through a system like the Gibco Rotea system. So 4 main advantages is low output volume. This is very important because, for example, you want to do electroporation. You don't want to electroporate, large volume, because then that you'll need more engineering reagents, right? You'll need more Cas9. You'll need more, guide RNA. So you can go, with the Rotea, you can go from, let's say, a liter of cell culture volume to 5 mils. So you can go as little as 5 mils with the Rotea. Process flexibility, that means you can do a lot of activities and a lot of applications with the Rotea without harming your cell yields and the viability. I am a cell biologist myself, and I call myself a cell culturist and a cell mommy. I don't like it when I lose my cells, and I don't like it when they're not happy. So with the Rotea, it's so gentle on the cells, so you have high cell recovery, and two, you have a high viability. You maintain the happiness of the cells. And last but not least, with this one machine, you can go from research through process development and all the way through patient reality or commercial manufacturing. All right, so what do you get when you purchase the Rotea system? So first is you get this compact instrument. I'm a very small person. I'm not even 5 feet tall. I have very short arms. I can hug the Rotea. That's how compact it is. So the footprint, again, is very important. A single-use kit, here you go, here is the consumables kit. 1, 2, 3, 4, 5, 6, 7 input and output ports. 8th port right here, that's what we call the H port or the harvest port. And then you'll get 2 pieces of software, the graphic user interface, or you can hear me call it GUI, and also the protocol writing software. And you'll get a laptop with a Rotea, and that's going to connect to the Rotea. It'll tell the Rotea what to do. Now, the protocol writing software, you don't need to write it using the laptop that connects to the Rotea. It does not have a software license, so you can put in any laptop that you want, and then put your protocol in a thumb drive and then upload it when you're ready to run the Rotea. Also, I want to make sure that you know that the GUI, or the graphic user interface, is so user-friendly, so please don't be afraid of it. Plus, also, we're going to train you, and we'll support you from the beginning all the way to the end, which means it's continuous, because hopefully you'll use the Rotea forever. So next, I want to show you here the process flexibility of the Rotea. So you can use it here. This is a typical cell therapy processing from vein to vein, and you could use it to thaw cryopreserved material and exchange it to media, or you can do the reverse. You can wash and concentrate from the actual cell culture volume that you have there, and then put it into cryopreservation media. So also, like I said earlier, you can also use the Rotea to wash and concentrate into an electroporation buffer to get ready for your genome engineering. We have protocols for each of these, so you're not going to have to reinvent the wheel. And like I said, we will support you every step of the way. How does the Rotea work? If you don't know about counterflow centrifugation, it's just how it sounds. 2 forces at work here: flow rate this way, G-force this way. Those are the 2 forces that are at play here and that the cells will feel. But the way the Rotea is engineered, the Coriolis effect is the pressure or the forces, how you feel, how the cells feel these 2 forces, flow rate and G-force. But because of the small chamber, it does not feel that Coriolis effect too much, and that's why it is very, very gentle on cells. So the cells go in this way. I call this the straw. My engineers hate it when I call this straw, but I'm a biologist. That's how it looks to me. So the cells go in, and when you control the G-force and the flow rate, or the mils per minute, you will dictate to the machine what you would like to stay, what cells you would like to stay in the chamber. And that is what we call the fluidized bed. So the fluidized bed, or the cells, it's kind of like a tornado. It's just going around and around right there, right here, and it's never a pellet. So with counterflow centrifugation, the cells are never pelleted. Like I said, it's just like a tornado. So with your control, so you control this, mils per minute and G-force, you will know what cells you want fluidized and what cells you would like elutriated. So what I mean by elutriations are the cells that are not in equilibrium with the flow rate and the G-force will elutrate out or exit out of the chamber, leaving the cells that you want in the CFC chamber. That's what we call elutriation. So for example, red blood cells, platelets, right? They're smaller than your lymphocytes and your monocytes, so you can elutrate out the smaller cells. Or Later on, I'll show you some other applications where you can use the Rotea to separate out other cell types. So here you see the cells that you don't want elucidating out because you're controlling the flow rate and also the G-force. Control is with you. So here again, it shows you, you load the cells. Here we go. You elucidate out the cells that you don't want, then you wash. Because it's never pelleted, you don't have to resuspend, right? You don't have to go up and down or anything like that. The cells will be washed very, very efficiently in the Rotea itself, and then you exchange to whatever buffer you want, whether it's electroporation buffer, cryopreservation, anything that you want. Now, you do have to be very careful that the density of the washed media is not very different from the density of the media that your cells are in, or else you will disrupt that fluidized bed. So again, we will train you with our protocols that go around that, and so we will help you as you're using the Rotea. And then you can concentrate the cells. So again, in the protocol, you will tell the Rotea, I want it to be concentrated into 5, 10, 20, however you want. Again, total control with you guys. So four main advantages again: process flexibility. You can do a lot with the Rotea. Happy cells. Third, low output volumes. And fourth, you can use it from research all the way through commercial manufacturing. This is one of my favorite slides here. I want to tell you that it's open architecture for process development, and then you can lock it down for GMP manufacturing. I like the fact that it's open architecture because I came from PD as well back in past life, and we will not charge you every time you want to change your protocol. That is your scientific creativity at work here, so we want to harness that, not hinder it. So we're not going to charge you for that. So it is open architecture, and then of course it's very important when you get to GMP that you lock it down. So that is why you can lock down the Rotea protocols once you are at GMP. So Rotea, is it GMP? Will it satisfy your GMP requirements? Absolutely. Here's the instrument. It's manufactured under ISO 13485. It is CTS, which means cell therapy systems. When you see within Thermo Fisher the CTS brand, you could breathe this sigh of relief because we take the regulatory burden, we minimize that from you, we take that from you so that you can focus on the technologies that you want to develop and get those therapies to patients. So it is CTS. The instrument is CTS. The single-use kit is also manufactured under ISO 13485 and in an ISO 7 grade clean room. Software itself, we have an additional software, an SAE system that you can purchase to make it 21 CFR Part 11 compliant. All right, so versatile, right? I said many different applications. I'm not going to read you this slide. Just remember Swiss Army knife of cell therapy. That is what the Rotea is. You can do a lot with it. Second favorite slide here because I love presenting data. So you can see here high cell recovery and viability over 95%. And then we concentrated also T cells 200 times with 97% recovery. And you could see the viability here still really great with greater than 90% recovery. And here you could see high PBMC recovery from a leukopak and only just a few RBC contamination here because there's still overlap, of course, with red blood cells and PBMCs. But you could see high recovery here. Then you could go on to your next step in the isolation of the cells that you want. This is how it looks like. These are 5 billion T cells in the chamber, so you got to watch out, of course. So it's continuous flow rate. There's no maximum volume really that you can process. It's just you got to watch the cells that are in the chamber itself. So you can harvest this out in loops. So you can, in one protocol, you can just do multiple harvests. And here's Ficoll versus Rotea. So you can see the growth of the T cells did not get affected. And also markers here. You can see Ficoll versus Rotea. There's no difference. So we did not affect the phenotype. Here's before CD8 as well, before and after Rotea processing. You could see that we did not change those ratios. And also we did not affect the phenotype because the Rotea is very, very gentle. Here again, Ficoll versus Rotea. Now Ficoll open, manual, very cumbersome, 2 hours. With the Rotea, you press that button and you're good. Less than 30 minutes to process the leukopak. This is, I believe, is a quarter leukopak. Many different cell types. This is a natural killer cells, a wash and concentrate. You could see the recoveries here and the viabilities and no changing of CD16 and CD56 phenotype, the markers. So you could process natural killer cells, Here you can clarify supernames when you're looking at lentivirus purification. These are 293 cells. Viral transduction right here, you did not change it, you did not affect it. It's because the Rotea, again, is very gentle on the 293 cells, which you don't need anyway. You could just harvest the supernames. And here are iPSCs processing, which my friends here from ViaCyte will talk about, and you can see the process development that we've done here when we were processing iPSCs. So last but not least here, I want to just say that we also are developing products so that it will connect to the Rotea, just like you know, these PBS and AIM5 in BPCs already. So these tubings right here will connect to the Rotea C-Flex and PVC, because the Rotea's single-use kits are PVCs. So we are constantly trying to make sure that you are the reagents that you need will be connected to the Rotea, because ultimately what you want is a closed manufacturing process. Right here is what we're currently doing, and also for our partners as well, is to connect the entire cell therapy process completely closed in a modular system, integrated but modular, under the control of a bioprocessing software like the TrueBio Delta V. So please let me know if you are, or your business development managers, if you're interested in partnering with us to make your processes completely closed. And with that, I thank you for your attention.
Thanks, Marianne, for the great introduction. Eric and I will now dive into ViaCyte's development of the Rotea for automated processing of our stem cell aggregates. So ViaCyte is a regenerative medicine company developing stem cell-derived cell therapies that hold the promise of a functional cure for patients with type 1 diabetes. Our cell therapy is a combination product, and so it's comprised of stem cell-derived insulin-producing cells encapsulated in a immunoprotective device. And so after implant, the device becomes vascularized to allow for nutrient and small molecule transfer across the membrane. And so our insulin-producing cells can then respond to blood glucose levels. As you can see by the graph here, we have performed preclinical studies that show successful control of blood glucose levels. The x-axis is time in days, and the y-axis is showing blood glucose levels. The green line is a non-diabetic mouse, and so you can see stable levels. And then the blue line is a diabetic mouse where you can see extremely large fluctuations. However, after implant and over time as our cells mature in vivo, they begin to respond to glucose levels and can effectively control them, similar to that observed in the non-diabetic mouse. And so the overall goal is to provide long-term insulin independency to diabetic patients. So our production process occurs over about 30 days. Initially, we thaw human embryonic stem cells and expand those in flasks and cell stacks over a period of 15 days, and to generate enough cell product to then aggregate and perform a directed differentiation down the pancreatic progenitor lineage. Upon the final day of differentiation, our pancreatic endoderm cells, or PECA1, are frozen down for long-term storage, and then upon need, they are thawed and seeded into one of our device configurations, where that device is then implanted into our patients. Worldwide, there is an increasing incidence of type 1 diabetes. So to support the millions of diabetic patients, we would require a commercial scale production of our cell manufacturing process. So to meet this demand, one option would be to scale out our process. Scaling out involves multiplying the number of batches. And so in our case, that would lead to hundreds of roller bottles. And as you can assume, that becomes unrealistic very quickly, as this leads to high aseptic risk, a large production footprint, and higher costs. Therefore, it's important to develop a scale-up model where we increase batch size to support large-scale production. So scaling up a cell therapy requires multiple considerations, a few of them being process comparability, so ensuring that across batches and across scales, you're producing the same product, you're maintaining product integrity. Additionally, it's important to establish a great relationship with your vendors and even consider multiple vendors for various consumables and reagents. And then additionally, being aware of the facility capacity needs and whether you perform your production process in-house or in collaboration with a CMO. However, the focus of today's presentation is really identifying scale-up technologies that meet the process requirements. So for our process, we have decided to transition to suspension bioreactor systems because they're closed, controlled, and scalable, which allows for a robust commercial production process. And so in conjunction with the bioreactor, we require a cell separation system to perform closed media exchanges and harvesting processes, which leads us to the Rotea. And so Eric will be discussing how we've incorporated the Rotea system as a suitable technology that meets our needs.
Thanks, Emily. So first, some details on the aggregates and the drug substance that we are using, that we're processing with the Rotea. ViaCyte cell drug substance, or PEC-01, consists of pancreatic progenitor aggregates. These aggregates are pretty dense. They're comprised of multiple cell populations at different phases of differentiation. There's also extracellular matrix in each aggregate. Usually we see aggregates between 200 and 400 microns in diameter. The aggregates typically grow in size as we move along the differentiation process. So, like Emily mentioned, we want to use the Rotea system as part of our aggregate production process. And the Rotea system is good for this application because it addresses several of our requirements for aggregate processing. Specifically, it allows us to accomplish media exchange, passage, and harvest of the aggregates and cells while maintaining a closed environment. It's also an efficient processing regime. The entire process should take less than an hour in total. There's no effect to the aggregate viability, morphology, or characterization after processing. Rotea is also a gentle processor, and its single use allows us to maintain a closed environment and minimize aseptic risk. It also allows the isolation of a specific cell population, so we can separate the aggregates from non-aggregates and also from any dead or single cells that we don't want in our final harvest population. And then finally, as Emily mentioned, it is a scalable technology, meaning that as we scale up our production process, the Rotea will be able to be scaled with us. So a brief overview of the PEC-01 scale-up development workflow and where the Rotea fits into that workflow. To start our production process, we expand human embryonic stem cells from thaw into 2D adherent culture on flasks, move from flasks to cell stacks, and then we aggregate our cells in preparation for differentiation into the final cell product. This differentiation process occurs in suspension bioreactors, and this is where we see the Rotea being integrated into our process, allowing us to have a closed process media exchange and harvest, and also efficient and streamlined processing to process our aggregates and remove any non-aggregate debris. After this part of the process, we then move to cryopreservation and post-thaw culture, which finishes out the production of our cell therapy product. So in order to integrate the Rotea into our process, we first had to perform some initial testing with our aggregates just to see what parameters would be good to maintain aggregate morphology, integrity, and viability as they were processed. And the way we did this was with just a very basic initial testing setup using a couple of bottle assemblies I'm going to walk you through here. And this allowed us to see what flow rates and what centrifuge speeds were best suited for maintaining aggregate morphology, integrity, and viability. So first, we drew aggregate cells in through an input bottle. This would be representative of our suspension bioreactor. The cells were drawn into the Rotea counterflow centrifugation cone, and we tried to form beds at various flow rates and centrifuge speeds. The spent media from the input is sent to the waste along with any non-aggregate cell debris that was removed from the fluidized bed by the Rotea. The aggregates still in this counterflow centrifugation cone were then washed with fresh media. And finally, the processed and concentrated aggregates were sent from the Rotea to a harvest bottle, where we could then confirm that the aggregates were still viable and still intact. And this setup allowed us to narrow down the range of flow rates and centrifuge speeds that best maintain our aggregate properties that we were looking for. So like I mentioned, there were two process parameters that we were primarily interested in optimizing. One is the pump flow rate, and the reason we're interested in this parameter is because if it's too low, we have a slow processing time with the Rotea, but there may be some improved aggregate retention. Conversely, if pump flow rate is too high, we get faster processing, but we might have lower viability or retention of our aggregates. On the flip side, when we're optimizing centrifuge speed, if the centrifuge speed is too low, you won't retain any of your aggregates. They'll be eluted directly to the waste. Conversely there, if your centrifuge speed is too high, you're going to have low aggregate viability, and you might also retain some of the debris that you don't want in your final product. So these were the two primary parameters that we were interested in optimizing using the test setup that we just saw. It is worth mentioning there are several other parameters that may affect the process inputs or outputs. However, these were assumed constant for testing for practical reasons. Some of these include the media density and viscosity, the density of the aggregates themselves, the diameter of the aggregates, and the number of wash steps performed by the Rotea during our process. So one of the other things that we were evaluating during these initial tests was we wanted to make sure that our process actually formed a fluidized bed in the counterflow centrifugation cone. And this is important because the fluidized bed maintains the integrity of the aggregates and also ensures that they are correctly retained and sent to the harvest as opposed to being sent to the waste. And as you can see on the picture on the left there, we were able to successfully form a nice looking fluidized bed in the counterflow centrifuge cone. We're also interested in evaluating the viability of the aggregates after processing, and this could be visually seen by any turbidity in the counterflow centrifuge cone during processing. And also post-process, we would look at the aggregates under a light microscope as seen in the middle picture there and confirm that the aggregates were intact, and then using software to make sure that our diameter after processing is comparable to the diameter before processing. We would also look at the viability before and after processing, as seen on the graph on the far right. Pre-test, we would see aggregate viability in the high 90%. Post-test, we would also see comparable viability after Rotea processing. Another thing we're interested in for our production process is processing the aggregates over multiple days over multiple media exchanges. And this is because, as Emily mentioned, our production process takes several days. So we want to be able to use the Rotea for multiple days if possible without any negative effects on our aggregates. And so during our testing, we processed the aggregates for multiple successive days in a row and observed a minimum loss in viability, as seen on the graph on the left here. Over 3 days, we would process the aggregates once each day with the Rotea, and we would see minimum change in viability after each processing at only about 3% loss in viability after each test. We also looked at diameter after multiple successive processing days, and once again saw a minimal change in aggregate diameter as seen on the graph on the right. In fact, on day 2, we actually saw a slight, not statistically significant, but a slight increase in aggregate diameter, suggesting that we're in fact removing some of the non-aggregate debris that would have small diameters. That's a good thing. We wanted to see that debris get sent to the waste instead of the harvest. So now that we've established the approximate initial conditions that we want to see integrated into our production process, we can also look into possible future applications of the Rotea to our cell therapy production process. So this is our overview of our production process: expansion, differentiation, cryopreservation of post-thaw culture. We're currently looking in the middle there to integrating the Rotea to our aggregation differentiation steps. However, in the future, we might be able to look to integrating the Rotea to our expansion steps, where the Rotea might have a role in stem cell expansion and cell passaging. It might also be useful for our cryopreservation and post-thaw culture steps, specifically for the cryopreservation harvest and during our post-thaw culture. And with that, some concluding remarks. ViaCyte is developing some stem cell-derived therapies to functionally cure type 1 diabetes. And in order to meet this ambitious goal, we need an ambitious scale-up effort to support our predicted cell therapy demand. The Rotea provides a scalable technology that can support closed and efficient processing of our stem cell aggregate therapy. And our preliminary results show high aggregate viability and normal morphology over consecutive processing days. Thank you for your attention.
Thank you, Marianne, Emily, and Eric for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, how can you process larger volumes, i.e. 10 liters, with the Rotea?
That is a good question. So as you scale in batch volume, your cell product volume increases as well. Since the maximum hold volume of the Rotea counterflow centrifugation is 10 milliliters of fluidized bed, the path forward would be to process the bioreactor solution in batches, each one comprised of a maximum aggregate volume of 10 milliliters. And this can occur multiple times depending on your load size. And additionally, the high flow kit allows for improved flow rates to support faster processing of these larger batches.
Great, thank you so much. And our next question, why choose a suspension culture method, i.e. in general, what are the overall benefits you see in suspension culture versus 2D adherent methods for stem cell research?
Yeah, so the advantage of suspension culture over 2D adherent methods for stem cell production becomes really apparent as the stem cell expansion and differentiation process is scaled. Compared to 2D adherent methods, stem cell production processes utilizing suspension culture combine improved cell processing characteristics such as unlimited capacity for growth, so no surface area limitations, and then at a lower aseptic risk with lower production costs, so reduced equipment footprint and labor requirements.
Thank you, Emily. Our next question. What led to ViaCyte's decision in selecting the Rotea for bioreactor processing?
Yeah, so in addition to its ability to maintain aggregate viability, integrity, and morphology, the Rotea's Counterflow centrifugation technology is scalable, making it suitable for ViaCyte's process as we scale up production. Additionally, the Rotea also matches our requirement for repeated batch bioreactor media exchange versus typical perfusion-based processes on the market.
And what are the steps planned to make it suitable for use in later phase and commercial processes?
So thank you for that question. I'll take this one. So it's the CTS Gibco Rotea System is designed for clinical manufacturing. So the instrument, the single-use kit, they're all CGMP grade and manufactured, like I said earlier, under ISO 13485. So the official regulatory message for the instrument and the single-use kits it's for research use or manufacturing of cell, gene, or tissue-based products. And also, like I mentioned earlier, the Rotea software has operator and administrator modes for manufacturing. And then the Rotea also has a 21 CFR Part 11 compliant software solution that will be available in the third quarter of this year. So thank you.
Thank you, Mary Ann. Our next question, is the entire product contact flow path for the CTS Rotea single use?
Okay. Question. So the entire fluid path for the Rotea system single use kit was evaluated for regulatory requirements. So that includes biocompatibility, endotoxins, extractables, and a lot more. You can see it in our COA. And all the regulatory testing results are available with the Rotea Systems Regulatory Support File, or the RSF.
All right, thanks again, Mary Ann. Our next question, can single-use kits be reused for testing purposes?
Yes, you can reuse single-use kits for testing purposes, but it is important to watch the integrity of the pump tubing and to clean out the tubing and counterflow centrifuge cone after each use to remove any debris or any adhered aggregates or cells. However, these kits are advertised as single-use, so the official recommendation for use with the final process is not to reuse the single-use kits.
Thank you, Eric. And our next question, what are the greatest challenges to Rotea optimization for aggregate processing?
Good question. The stem cell aggregates that we work with, they require gentle handling because they can be quite fragile. It's necessary to maintain aggregate integrity during the processing because this is a critical part of their functionality in the final product. So modifying the Rotea process parameters to adequately collect, wash, and refeed the aggregates from the bioreactors, it took some fine-tuning to do, but assistance from Thermo Fisher and working through the parameters to optimize the shear forces was an interesting process. However, we have achieved successful cultures utilizing this system.
Thanks again, Eric. Our next question here is, can you further detail other potential areas of ViaCyte cell process where the Rotea could add value?
Sure. So as we discussed in the presentation, ViaCyte has adapted the Rotea to our stem cell aggregate differentiation process. However, we see a multitude of other areas where the Rotea could offer support. One of those includes the final harvest for cryopreservation. When we generate our drug substance, we require an intermediate freezing step for long-term storage, and the Rotea could benefit this process by concentrating, washing, and collecting the final product in cryopreservation media.
Thank you. Our next question, when adapting your feeding and harvesting processes to the Rotea, did you run into any issues if the aggregate solution is not uniform, i.e. heterogeneous mix of smaller to larger aggregates?
That is a great question because our aggregates do tend to range in size. We commonly see aggregate solutions that have several standard deviations worth of size, approximately 50% there. But since the aggregates are so much larger than the single cells we want to remove from the solution, we haven't had a whole lot of issues separating the two.
Thanks again, Eric. Our next question is, how does the CTS Rotea compare to the Prodigy from Miltenyi?
Okay, I'll take that one. Thanks. I get this a lot. So I'll answer this question. So the Rotea is very different from the Miltenyi Prodigy on a lot of fronts. The Rotea, again, I had my favorite slide. I'm going to quiz you on this now. My favorite slide, right? I said that it's an open architecture. So it's an open platform, that it's reagent agnostic and bead agnostic. So you can use your whatever reagents that you would like. Like I said, it's open. You have the control whenever you use the Rotea. So, and also I showed you the vision that we have, so you can combine it with, offline incubator scale that's better for manufacturing because it doesn't have an integrated incubator like the Prodigy. So it allows for modular manufacturing. Thank you.
Thank you, Marianne. And our next question here is, how involved is the support for developing a process? Can you help design the program used for our cells?
Okay, I'll take this one as well. Thank you for that question. So Thermo Fisher Scientific, we are very invested in the success of each of our Rotea System customers. So we provide support throughout the instrument buying experience. So for example, a custom protocol is developed for each Rotea System demo in order to suit your specific application. And then a live cell demo is performed with the Rotea System for your evaluation. Also, we include initial equipment and software training by our trained field application scientists. And then post-sale, you have the entire support system, myself, the field application scientist, and the tech support teams are available for further protocol development assistance. So we're all here for you. Thank you.
Thank you. Oh, thank you, Marianne. Our next question here is the system often used in combination with cell factories, e.g. media exchange, and also CF40?
I'll take this question, and thank you for asking this. Yes, so the Rotea system is it's so tremendously flexible, and it's designed to be completely customizable. So it's been connected to incubators, bioreactors, CF40s, cell factories, as part of a closed manufacturing system. Like that one slide, that last slide that I showed you, that is the ultimate goal, is to have a entirely closed manufacturing process. So in order to connect to cell factories, what we use is off-the-shelf PVC C-Flex adapters. So that's required. Thank you.
Thank you. And our last question here, what is the approximate process time to go from 1,000 milliliters to 5 milliliters?
Marianne again here. So depending on the chosen settings, again, you have the control. The volume reduction processing time for 1 liter down to 5 mils can be, as fast as 20 minutes. Thank you.
Great. Thank you again, Marianne. Thank you again, all of you, Marianne, Emily, and Eric, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Gibco by Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.